amino acids are going to be the building blocks of our proteins in our bodies. Now, in total, there's 20 different amino acids. We're not going through all 20 of them, don't worry about that. But all 20 of them do share a common trait. They all have the same basic chemical structure. So if you're looking at this image here, you can see there is a central carbon. Now, that central carbon is always going to be there. Then next to that, you'll notice there's this amino group. The amino group is a nitrogen with 2 hydrogens attached, so NH2. That's always there. You then have the carboxyl group off the other side. Now you can see it's a carbon double bonded to an oxygen, and then you have the same carbon bonded to an oxygen bonded to a hydrogen. So COOH, a carboxyl group. Now that takes up 2 of the 4 bonding sites so far from that central carbon. The third bonding site is simply a hydrogen, just a basic simple hydrogen. All right, that's 3 of the possible 4 bonding sites. The 4th bonding site off of that central carbon is this R group or radical group. That's where the differences occur across all 20 amino acids. Now, you might have a very simplistic group here, just a hydrogen. Others are huge ring structures. It really depends on the individual amino acid as far as what's in that radical group. But every one of the 20 amino acids have the same basic structure. Central carbon, amine group off one side, carboxyl group off the other side, hydrogen, and then the radical group, what makes it unique for each one of the 20 amino acids. But an amino acid itself doesn't make up a protein. You have to put the amino acids together. You need to make sure you build a chain, a structure of amino acids. Now here we're seeing, you can see in the box here, OH and H. This is how we will form what's called a peptide bond. What will end up happening is that HOH or H2O water will be removed. It's a process called dehydration synthesis. You're dehydrating, removing water, synthesis, putting together. So dehydration synthesis removes that water and builds the peptide bond between that carbon and that nitrogen. And this will happen over and over and over again. You'll build long chains of amino acids. Each one is going to bind by removing water, dehydration synthesis. Next one, dehydration synthesis, over and over again. Now, as you keep building these long chains of amino acids, that is what is referred to as the primary structure. The primary structure of the 20 different amino acids is just a long line. At this point, that's not an actual functional protein yet. Instead, what happens is that primary structure ends up being put into an alpha helix or these beta pleated sheets. That's your secondary structure. You might even combine the two in the same one. But from there, from that secondary structure, it goes into a tertiary structure. You continue folding and pulling it on itself. You're trying to create a particular shape to make this protein functional. And from that tertiary structure, quite often you go into this quaternary structure. Now in this case, this quaternary structure took several different, in this case 4 different tertiary structures and put them together. What ends up happening is this quaternary structure happens to be hemoglobin. Hemoglobin, the protein that's found on your red blood cells, or should I say in your red blood cells, that allows oxygen to bind. But it's only functional once it's gone from primary to secondary to tertiary to the final quaternary structure. Then that's a functional protein. So these amino acids, they can start the process, but then have to have all this folding and moving and rearranging into a final functional shape. So these different levels of proteins. But once you get to this final functional state, what do proteins do? Well, there's quite a few different functions of your proteins. First off is structural. Your proteins help to make structures in your cells. They help structurally in your bones. They help structurally in your muscles. They're helping pretty much structurally throughout your entire body to hold things in place, to give strength to things. So we have a lot of structural components that are based off of protein. Enzymes. Now, enzymes are something that's meant to help catalyze a reaction, help make a reaction occur quicker or easier. The great thing about enzymes, they can be used over and over and over again. They just keep on working. So here's an example we're looking at. The enzyme you can see is the base part. You can see there's kind of like two almost cookie cutter structures right here. Things will fit in there, but they're a particular shape or a particular chemical signature. You can then have S1 and S2, the substrates, the pieces. They come together, they fit into here, and then you have this structure, the two substrates being held together. The enzyme is facilitating bringing these two pieces together, because otherwise they're simply floating around inside your cytoplasm. That then ends up making your product, and it's released. So you've created this new structure from these two substrates. But if you notice, the enzyme is exactly the same. It didn't change. You can find two more substrates and use the same enzyme again and keep building these products over and over. It's helping to facilitate a reaction. Sometimes it's helping to facilitate the reaction by lowering the activation energy. So if you're looking here on the left one, you can see this large curve goes up and back down. That's how much energy is needed to create this product. Now with an enzyme, you might be able to lower this activation energy. So you don't need quite as much energy for it to occur. Therefore, making the reaction occur easier. You don't need as much energy, so therefore it's easier to cause this reaction to occur. So we can help facilitate the structural builds. We can help to lower activation energies. Enzymes can be very, very functional in your body. All right, next one is hormones. Various hormones in your body are made from proteins. Now, hormones, those are the chemical messengers that kind of float throughout your bloodstream. It's one of the ways your body signals different reactions to occur. So you're going to have the endocrine system producing hormones, and some of those hormones are protein-based. Contractile. Think muscles contract. Inside your muscles, you're going to have proteins that help facilitate the contraction. These proteins are allowing your muscles to contract, allowing your body to move throughout the day. So contractile. Immune proteins. Think immune system, fighting off infection. Immune proteins are going to help facilitate your body's way of destroying foreign particles. Could be viruses, bacteria, could be whatever, parasites, you name it. But these immune proteins can help your body fight them off, keeping you healthy. And transport. Well, we already mentioned hemoglobin. Hemoglobin's in your red blood cells. It helps transport oxygen. Helps sometimes transport carbon dioxide also. So transport, and that's just one example. There's many examples throughout your body of proteins helping with transport mechanisms throughout your blood, throughout your cytoplasm, through all your tissues. So we have transport as a mechanism. All right, let's shift into the fourth of the macromolecules here, the nucleic acids. Now, nucleic acids, you might have heard of these with DNA or RNA before. They're composed of carbon, oxygen, hydrogen, nitrogen, and phosphorus. So we're adding in this phosphorus component now. So a little bit more complexity as far as what the actual pieces are. Now the base units of your nucleic acids are going to be nucleotides. Now these nucleotides, in total there's 5 between the DNA and RNA, but these nucleotides will have a nitrogen containing base. That's kind of the base part of it. You're going to have a sugar, this is saying a pentose sugar, and then a phosphate group. Now, when you start looking at basically nitrogenous base, you're going to have a sugar and you have a phosphate group. So let's look at the actual structural components of these different pieces. Now, the full piece right here, it's only 1/2 actually, but that's the actual strand of DNA. So what we're going to do though is look at the building blocks, the pieces that make that up. So we'll start with the nitrogenous bases. Now look at the ones that are in DNA to start with. In DNA, you compare A and T, C and G. So first off, you have adenine, that's A, paired with thymine, T. So again, in DNA, this is how it's paired, AT, adenine, thymine. Then you also have C, which is cytosine, paired with G, which is guanine, CG. That's again in DNA. But if we switch over to the RNA, in RNA, we don't have thymine. There's no T at all. Instead, you have uracil. So in RNA, you have uracil, which replaces the T. So again, each one, DNA and RNA, only have 4. In DNA, it's ATCG. In RNA, the T is swapped with the U. From there, those nitrogenous bases will be added to a sugar, and then you have the phosphate group here. So all together, all 3 pieces are making up this structural unit, this nucleotide. You can see that you have a nucleotide, another nucleotide. Each one of these I'm circling is a nucleotide. You can notice that throughout all that, it's the phosphate right here that's linking them, creating this kind of phosphate sugar backbone. With the actual nitrogenous bases are the ones that are in toward the middle. So you're looking at the building blocks, the structure of DNA and RNA for that matter. When we put it all together, it's a double helix. You can see here how you have adenine is red and you have thymine, which is yellow. So you have all the red-yellow pairing. You'll also notice guanine is blue and then cytosine is green. So all the C and G pairings. Those then have this outside strand that you're kind of tracing down. That's the backbone. That's the phosphates and sugars. All the bases are what are right in the middle here. The bases bind to each other, creating this kind of ladder, this kind of building structure in the middle of the DNA. RNA, pretty much the same. I've already talked about this, how it has the 4 different bases. Now it uses uracil. But RNA is single-stranded. So we just saw DNA is double-stranded. RNA is single-stranded. So while DNA is deoxyribonucleic acid, RNA is ribonucleic acid. Now this single-stranded uses uracil, as we mentioned, but now we have different types of this. We have mRNA, we have tRNA, we have rRNA. Each one has a different function. mRNA being messenger RNA, then we have transfer RNA, we have ribosomal RNA. Each one has its own purpose. So while they're all single-stranded, they're found at different places and different functions throughout our cells. And now I'll bring up ATP, adenosine triphosphate. Well, why is that here? Because ATP, adenosine triphosphate, still uses a nitrogenous base. You have adenine here, same thing as DNA and RNA. And then you have this ribose sugar and the 3 phosphate groups. The energy that is ATP, and this is our energy source for all of our cells. The energy is what's stored in these bonds. There's more energy in the attachment of all 3, so this last one has the highest energy. But even adenosine diphosphate, so if we cut it right here and only have the 2 phosphates, there's still energy, just not as much. So ATP, adenosine triphosphate, uses the backbone, adenosine, just like DNA and RNA can use. But this now is going to be a main energy source of our body. So the idea of proteins, the idea of nucleic acids, 2 of the 4 main macromolecules we find in our bodies.